So the third group we're going to talk about are the proteins. Now, proteins are the most common organic molecule we're going to see in cells, and they're super important. Proteins do really most of the functions in a cell. They have regulatory functions, they have structural functions, they protect the cell, they move stuff around the cell, they break stuff down in the cell. They're really the molecule that's doing things for the cell. So we have a special name for all the proteins in a cell combined together. So if we're talking about all the proteins that a cell makes, that it has, we refer to that as the proteome. Now you might have heard of the genome, which is the genetics of a cell, so the DNA. I'll talk about DNA in a second. And the proteome is really equally as important because proteins take action. So when we're looking at proteins, we're going to be focusing on two different functional groups. So we're going to see a carboxyl group, which we've talked about before. We saw the carboxyl group when we were looking at our triglycerides and our lipids. We're also going to see an amino group when we talk about proteins. So this amino group is specific to proteins. It's going to be really important. The structure of a protein starts with the monomer. The structure of a protein starts with something called amino acids. So amino acids are going to be our monomer for proteins. Now, there are 20 different kinds of amino acids. We're not going to learn about all of them. What you should know is that they have different chemical and physical properties. The fact that there are 20 of them is really why proteins can do so many different things. There are some commonalities between them. So I do expect you to know this, what's on this slide, the different parts of an amino acid. Okay. So every amino acid has a central carbon. So there's a carbon in the middle. It's called the alpha carbon. Right below that central carbon, there's a hydrogen. We don't need a group name for that. That's just a hydrogen. But to the left and to the right, these two groups are really important. So we have our carboxyl group here on the right, and we have this amino group here on the left. Okay, so we can see that the amino group contains nitrogen. The carboxyl group here is carbon and more oxygen. So those are going to be in every amino acid, the alpha carbon, the amino group, the carboxyl group, and this little hydrogen. What's going to be different is here where it says R. So this R, our R group, is a variable side chain. So just like when we were talking about steroids, I mentioned that there's an area of the molecule that can vary. This R group in amino acids varies and gives those amino acids their function. So this could be anything. Except not exactly anything. It's going to be one of these things. So there are 20 choices for amino acids, and you don't need to know them, but I want to highlight here for you that some of our amino acids are going to be non-polar. Some of them are going to be polar, so we should realize that some of our amino acids are going to repel water. Some of them will be attracted to water. We have positively charged groups, negatively charged groups, both of which would be attracted to water, right? Anything with a charge is hydrophilic. We'll have some non-polar R groups that happen to be in the shape of a ring rather than little chains. Okay, so you don't need to know them, but you should know that they have different chemical properties. Okay. The other thing that allows proteins to do so many different jobs is that proteins have complex structure. And what I mean about that is that they're going to have three-dimensional structures. They're going to be kind of crumpled in on themselves in a huge variety of ways. So when we're talking about proteins, we kind of specify what level of structure or folding we're talking about. So we're going to go through it right now, and we're going to have 4 levels of structure for proteins. Now, the first level of structure is going to be taking our amino acids, our monomers, and forming a polypeptide. So our polypeptide is our polymer for proteins. Okay, our polypeptides form when we make a peptide bond between two amino acids. So a peptide bond is a covalent bond, just has a special name. It's the covalent bond that you find between two amino acids when you're making a protein. The peptide bond forms between the carboxyl group of one amino acid and the amino group of the neighboring amino acid. So we can see both these amino acids, glycine and alanine, have an amino group. Here we see an amino group. Here we see an amino group. And both also have a carboxyl group. Here's a carboxyl group. Here's a carboxyl group. And their R groups would be here and here. Okay. So when we're linking them together, we take the carboxyl group on one of them, in this case the glycine. We take the amino group on the other one, and we link them together, that carbon and this nitrogen, using a hydrogen bond. Okay. As we do this, water would come out, so this would be a condensation or dehydration reaction. But the important thing we want to note here is that as we're forming this larger polypeptide, this protein, this is going to affect what we see in this chain that we're forming down the center. So because each of these amino acids has this amino group, and we're linking the carbon of the carboxyl group to the nitrogen in that amino group, if you look down a chain of a polypeptide, if you don't look carefully, you might think it's just carbon, carbon, carbon. So you might get confused. You might think, is this a fatty acid or like a really long linear carbohydrate or something? No. What we're going to notice as we look here is that what we're actually seeing is nitrogen, carbon, carbon, nitrogen, carbon, carbon. And that happens all the way down our polypeptide chain. And that's very, very specific to proteins. If you see this nitrogen, carbon, carbon, nitrogen structure, that chain, you can only be looking at a protein. You know you're looking at a polypeptide. That's because these two carbons form from the central carbon, this carboxyl group, and then we always, always, always are linking together to our next amino acid, which is always going to link in starting with this amino group. The other thing we can recognize about polypeptides, other than this chain that we're forming of carbons and nitrogens, is that we're going to have a lot of variable side groups, these R side chains coming off. So this is because we have our 20 different amino acids, and in blue we have highlighted the R group for each of the amino acids that is creating this specific polypeptide. The other thing we can notice about this polypeptide that is based on the structure of that amino acid, so we have an amino group on one side, a carboxyl group on the other side of every amino acid. When we link them all together, that means at the end of a polypeptide, we're going to have an amino end and we're going to have a carboxyl end. So every polypeptide is going to start this chain really with a group containing nitrogen, ignoring the hydrogens, and we're going to end with this free carboxyl group, this carbon linked up to oxygen. So we're always going to see that if we're looking at the chemical structure of a polypeptide. Okay, so that's what forms our primary structure of a protein. So the primary structure is just the amino acids linked together. So here we have shown our amino group from that first amino acid. Here's the carboxyl group from that last amino acid. And here we have abbreviations for each of the amino acids here. So we didn't write out the specific chemical formula, but these beads, each bead is an amino acid. So that straight structure is our primary structure, and it's held together by peptide bonds. I'm specifying that here because each of these levels of structure is going to be created by a different type of bond or association between the elements, between these functional groups that we have in our protein. So the secondary structure of a protein actually forms only really one of two specific shapes. So the secondary structure of a protein is going to form because we're going to create hydrogen bonds here, hydrogen bonds between the hydrogens coming off of our nitrogen in that amino group and the oxygens in that carboxyl group. So remember, whenever we have oxygen here, we have a slight negative charge. This hydrogen happens to have a slight positive charge. So this is going to cause the primary structure, that polypeptide, to fold over on itself and allow hydrogen bonds, which remember are just kind of like a little like magnets. So we're not forming a covalent bond. We're not sharing electrons. We're just kind of sticking together because they're attracted to each other. So these hydrogen bonds are going to form. And this creates one of two shapes in any part of a polypeptide. So our first one, alpha, because scientists like to label things just A and B. So alpha and beta, we're going to have an alpha shape and a beta shape. Our alpha shape is called the alpha helix. So a helix is like a swirl, a spiral, a ringlet. So in an alpha helix, these hydrogen bonds pull our polypeptide into a swirled spiral structure. Our second option for a shape we can form that's a secondary structure is our beta pleated sheet. So a pleated sheet, just like a pleated skirt, it's going to be kind of flat, but it's going to have a couple bends in it, still held together by those hydrogen bonds. We're also going to have a tertiary structure, so our third structure for a protein. Now, you'll note here that from our primary structure, so just our amino acids in a chain, we had our secondary structure, the alpha helices and beta pleated sheets forming because of the hydrogen bonds. Now, as we go into the tertiary structure, we can actually see that this polypeptide was pretty long. So some parts of it formed an alpha helix, other parts of it formed beta pleated sheets, and there are sections that just didn't really do anything, so kind of linked between them. Okay, so any given polypeptide, it's not going to form into necessarily only a helix or only a pleated sheet, but specific sections are going to form into each of those. Now, in the tertiary structure, what we start to have happen is these just crumple up. So that secondary structure, we had really specific shapes that we saw, the helices and the pleated sheets. And now they start to crumple up just like a ball of paper. So when we start to see an actual 3D kind of complex crumpled structure, that's when we're looking at our tertiary structure. The final level of structure that we have, our quaternary structure, so quaternary just means fourth, is when we have many different polypeptides interacting with each other. So in our quaternary structure, here we see our original polypeptide from before, right? So we've actually been tracking this polypeptide. So we tracked it into its primary structure, its primary structure, its secondary structure, its tertiary structure. Now we can see it participating in this quaternary structure. But in the quaternary structure, it's going to link up together with other polypeptides, each of which have their own primary, secondary, tertiary structure. And they're going to be kind of binding on to each other to form an even bigger protein. So that's our final protein shape, is when we have multiple, what we call subunits linking together. So I love tables, so I made you one. So this shows you the level of structure, primary, secondary, tertiary, and quaternary. Tells you the shape. So the primary structure in a protein is a chain. The secondary structure is an alpha helix or a beta pleated sheet. Tertiary is some crumpled 3D shape. And a quaternary structure is multiple subunits built from multiple original polypeptides. And then below we have the type of bond that creates this level of structure. So our primary structure is the peptide bonds between amino acids. Our secondary structure is hydrogen bonds forming to pull us into these shapes. I didn't mention this, but our tertiary structure, the balled up kind of crumpled sheet of paper structure that forms, it forms because of the interactions of R groups. So we can't be more specific than that because we remember that our R groups are variable side chains. So we don't really know what they are. They're going to be different for every polypeptide. They're going to be in a different order for every polypeptide. So that's why we can get so many different tertiary structures. So every different protein, every polypeptide is going to crumple up slightly differently. And finally, our quaternary structure kind of also depends on those R groups, but specifically, it's going to depend on the properties of the R groups that are like sticking on the outside of the tertiary structure of each of the subunits. So these subunits are going to have, like, I don't know, maybe they'll have a polar part here, a polar part here on two neighboring subunits. And so then they'll kind of magnet together, right, because they're attracted to each other, something like that. So interactions between our two or more subunits. So an example of why we kind of care about these levels of structures in proteins is sickle cell anemia. So sickle cell anemia is a disease that affects hemoglobin, which is a protein in red blood cells. And it actually happens just because of a mutation that creates one difference in your primary structure. It causes a substitution of one amino acid in this original chain. But that error kind of like propagates. So propagates just means continues on, kind of like dominoes. So this error, as we follow it through these levels of structure, what happens is that in this primary structure, we fold into our alpha helices and beta pleated sheets, and then we've combined this also with the crumpling of that sheet of paper due to R group interactions into a tertiary structure. So normal hemoglobin, and this subunit forms into this kind of globular shape, right? Looks a little like a heart maybe. But in sickle cell hemoglobin, if we have this mutation, we form into this shape that has this sort of depression in it, right? And a difference here is important that we now expose some hydrophobic R groups. So some water-fearing R groups are exposed here in our sickle cell hemoglobin that aren't exposed in normally functioning hemoglobin. And that's going to affect how our whole red blood cell works, really. So this is one of our subunits. And when we have our subunits linked together in normal hemoglobin, it would look like this. Sickle cell hemoglobin, it would look like that. Now, hemoglobin molecules usually won't associate with each other, and each one will carry oxygen when hemoglobin functions normally. But when we have sickle cell hemoglobin, because we have these hydrophobic regions exposed, and because we have this new shape, the hemoglobin molecules actually link together. They do interact with each other, and they kind of crystallize, which really impacts how much oxygen they can carry. So they can carry less oxygen because they're kind of linked together, so they don't have the same space they would usually have. So this is a way that the shape of proteins really matters, really affects how something in the body functions. Now, a special protein type that I want to highlight for us before we finish this section of our macromolecule class proteins are our enzymes. So we're going to talk about enzymes more when we talk about metabolism at the end of this course. But I want you to note that enzymes are a type of protein, and they're a type of protein where, again, the structure, the shape of the protein really matters. So the way an enzyme works is that it has a specific shape that binds into a specific molecule, some other molecule, has this shape that fits another molecule somewhere in the cell. And the fact that the enzyme and this other molecule fit together lets this enzyme do stuff. So specifically what it's going to do is catalyze other reactions in the cell. So it's going to speed up various chemical reactions. And it does that because it can bind on to other molecules in the cell. And then the enzyme can change shape slightly. So it can take that molecule, for example, and break it into two pieces by kind of stretching it, right? If we stretch this section, we could more easily break that molecule in half. And this is one of the ways that we're going to get cells to do lots of a chemical reaction that otherwise would happen really slowly or wouldn't be likely to happen spontaneously. So this is really important in human biology and also the biology of other organisms.